Hello friends, in this video we'll be seeing the translation DNA synthesis. No, it is not translation, TION, it is translation DNA synthesis. Lesion means segment of the DNA which is just get hampered due to any kind of modification. It could be a mutation or it's an indication of the mutation. Normally these are the chemical modification on the bases, like here we can see it is a modification of thymine bases. When two thymine bases come together they form a dimer of the thymine. It is due to the UV radiation, sometimes the X-ray radiations too. Now this kind of thymine dimer formation can lead up to the disrupted structure of the DNA backbone. It will make a kink like this. Now as a result, it is pretty difficult for replicating a DNA onto the opposite side of this strand. But still, this task have to be done sometimes. And most of the times, actually, these thymine dimers are repaired. Sometimes they are repaired utilizing a system which is called photoreactivation with the help of the enzyme photolase, which will cleave this thymine dimer and insert which will just disrupt this thymine dimer and make those thymines just sit together, not as a dimer but individual nucleotides. But there are also repair mechanisms like base excision repair system or nucleotide excision repair system, which will just cleave off few nucleotide stretches behind and beside this thymine dimers and kick this out and synthesize it with new DNA strand. But in this case what we can see here are the new replication procedures where they just replicating this thymine dimer containing template without deleting this thymine dimer, without repairing this thymine dimer. So they are just carrying out the DNA replication process with having this defect onto the DNA. Okay, so this can also be possible because sometimes there are a lot of mutations are going on, and if cells are making all the ingredients to do this, that is really difficult, and in those situations cells sometimes need to carry those mutations with themselves, and this is called the Sos repair system, right? So it's also part of the Sos system when you have nothing to do because you are in an environment which is so much changing, where there are a lot of radiations are coming all the times. So in those situations you cannot do all this repair mechanisms, you cannot rely on those repair mechanisms. So sometimes you need to go with the changes in the DNA, okay, like the thymine dimer. Okay, so in this case our DNA polymerase 3 will come and attach to it, and it will start synthesizing with the help of the beta clamp, which is thus increasing the processivity of this DNA polymerase 3. Now right after the synthesis, when this part or this kink region of thymine dimer comes for this DNA polymerase 3, this polymerase will fall off because this conventional DNA polymerase does not have the ability to synthesize the nucleotide sequences against the template strand which is having this thymine dimer formed. So in this case they will need other type of a specialized DNA polymerase like Pol4 or Pol5. Now this Pol4 and Pol5, Pol4 is called the DIN B, which is a different protein, and Pol5 is called another protein which is UMU-D prime 2C. As we know, these proteins are also part of the Sos repair. So this is what we call the Sos, means you are just accepting the damage inside your DNA. So you cannot repair it anymore. You just take it, okay, fine, you have a damage, we go with the damage anyway, but we cannot stall replication because it will stop making the mRNA, it will stop making the proteins which is necessary. So just having the mutation and making it. Okay, that's what the goal of the Sos repair. Now in this case the Pol4 and Pol5 both are acting, any one of these can act at this position. Pol5 for example come here and attach to that, and it will start synthesizing or adding nucleotide sequences, though there are thymine dimer formed in this template strand, but that doesn't matter for them. They are specialized for doing this, and they can do all these things that actually don't bother for them. Okay, so you go through the strands and they'll synthesize the whole DNA sequences. That's how a mutation is being adapted. So it adapts a mutation. Whatever mutation is happening, we cannot do anything anymore. We don't have the necessary ingredients, so we have to go with it, and they are going with it. Okay, so that's all about the translation DNA synthesis, or it's a part of the Sos repair but not actually the overall Sos repair, and I hope it will help you. Thank you.